Hello everyone, my name is Yasser Mohseni and I'm a PhD student at Sorbonne University. I work in the Laboratory of Computational and Quantitative Biology, ECCB, under supervision of Professor Alessandra Carbone and Professor Elodie Lane. Today, I want to talk about our researches on the prediction of the effects of mutations on the protein-protein interactions with geometric deep learning. I will first introduce our objective of the project and the challenges we faced on the way and how we addressed them. Then I will continue by describing the database that we used for our methodology, which is combination of geometric deep learning and local structural variations. At the end, I will present our experiments and results. The objective of this project is to predict changes of binding affinity between two proteins upon mutation. Protein-protein interactions drive many biological processes in living organisms and are necessary to the cellular machinery. Disease-causing point mutations on a protein interface affect the protein's ability to interact with its partners in the cell. Hence, it becomes crucial to develop systematic and accurate approaches, assessing the impact of mutations on the formation and stability of protein complexes. Here is an example of a protein complex in which a point mutation happens on the interaction site. In this mutation, alanine is replaced by tryptophan. Because they both have hydrophobic side chains, this mutation only affects the geometrical properties of the interaction. However, if we substitute alanine with glutamic acid, which is a negatively charged amino acid, then the chemical properties of the interaction site will be changed as well. These changes manifest themselves as the changes in the binding affinity, which we represent by delta delta G. In this project, we faced two challenges. First is the reconstruction of the mutant protein complex after mutagenesis, and the second is identification of fine descriptors for the prediction of changes in binding affinity. Following single-point mutation, the side chain orientation angles of the mutant amino acid are unknown. Furthermore, the backbone of the protein is subject to slight movements in response to point mutation-induced changes. Therefore, we need to consider the effects of the mutant amino acid on the backbone torsion angles. We use Rosetta Backrub to explore near-native conformational fluctuations of a protein and to compute an enhanced estimation on side chain orientation angles of mutant amino acid and on backbone torsion angles. Using protein structures and information of point mutations from ECCB version 2, we created a database of conformational models for both wild-type and mutant structures. This database contains 208 protein complexes and 3,050 mutations. For each mutation, 30 wild-type and 30 mutant models are generated. In this process, only reliable binding affinity measurements are chosen. We analyzed the experimental delta delta G based on the regions of the protein interaction on which point mutation happens. According to Levy, the interfacial residues can be classified into three regions: support, core, and rim. As it is shown in the right figure, support is the most buried region, while rim has the highest solvent accessibility. As we can see in this figure, the mutations happening on the core have the biggest influence on the complex free energy. Therefore, it's important to know on which region mutation happens and integrate this information as an input feature for our deep learning algorithm. This slide shows how the conformational models are created for each protein complex of ECCB. Each complex goes through global optimization of backbone and side chain torsion angles. Then a local conformational sampling is applied on the interaction side to create 30 raw models. These models go through a parallel pipeline, one for wild type and the other for mutant, to apply discrete combinatorial rotamer optimization of side chains, also known as repacking. Given the importance of local structural variations for a direct estimation of binding affinity changes upon mutation, we implemented an end-to-end Siamese convolutional neural network that takes as input the local 3D environment around the mutation site in the wild type and mutated form and gives delta delta G as output. Thanks to the use of locally oriented frames, the architecture is invariant to 3D translations and rotations. The local 3D environments are extracted from conformations generated with Rosetta backdrop. Unlike other approaches that calculate delta G of mutant and wild type separately, we focused on local changes and differences between structures by analyzing the residues of interest on which the mutation happens. We placed a sufficiently large cube around the wild type and mutant residues to capture local information on how the mutation affects the interface. We also integrate evolutionary information and predicted mutational effects extracted by JET2 and GEM respectively. We separated training and test sets so that they share no common sequences. We considered 71 complexes with 1,417 mutations for the training process and 35 complexes with 370 mutations as a blind test set. We evaluated the performance by filtering test samples based on various levels of sequence identity. Here we have the performance as Pearson correlation on various levels of sequence identity. Samples of a given level of identity can be considered as samples of higher levels. Thank you for your attention.